proteins, right? So proteins are one of these organic compounds in the human body, right, that we need to talk about. So the monomer, or the single unit of a protein, or the other term is a polypeptide, but we'll talk about that, is an amino acid, right? Amino acids, there's 20 amino acids. Depending who you talk to, there's 8 or 10 essential amino acids, meaning we can't make them in our body, but some we can definitely make in the body. Now, a polymer is a protein, or we can call it a polypeptide. We call it a peptide because the bond we form between amino acids is called a peptide bond. So, proteins are used for all kinds of important things, like we talked about in class. So, enzymes, which are catalysts, are made of protein. Um, you know, Plasma proteins floating in the blood, including clotting factors like fibrinogen. And albumin is the main plasma protein. Also, proteins in the cell membrane, like transporters and receptors, where things bind to, or we pass ions in and out of the cell. Also, we use proteins for things like structural stuff, like collagen, right? Collagen in tendons, ligaments, fascia, and also in the dermis of the skin. And then we have contractile proteins, proteins that create movement. How do you create movement? Proteins, like in skeletal muscle, are called actin and myosin. And we have them in the muscles in our skeleton that move our skeleton around, like our biceps. And we have them in the heart. And we have them in this other muscle called smooth muscle around the stomach and esophagus and uterus and bladder and the blood vessel walls and the airways, all over the place, right? So if we're creating movement, it's with protein. If we have these structural things like tendon, ligaments, the dermis of the skin, which provides just tensile strength to the skin, it's going to be these proteins like collagen. And even the elasticity of tissues, we have proteins called like elastin, right? And that goes beyond the scope of this test, but we'll have to know it here in just about a week or so. So we'll have to go over all that. Okay, so tons of functions for proteins, and we know they form very specific structures, and that was what we called, that primary, secondary, tertiary, and quaternary structure, where the primary structure is just the chain of amino acids, and then it folds into these beta pleated sheets and alpha helices, and then it folds and bends more. And then the quaternary structure, the fourth degree structure, is where different polypeptides come together. We'll see that in things like hemoglobin. Okay. Now, the structure of amino acids here, you don't need to draw this formula out or anything, but that's what it looks like. We have this, which is the carboxyl group. So this is the carboxyl group. So we have carbon double bonded to oxygen, carbon bonded to an oxygen and hydrogen. Then this is the other end, the amino end, where we have a nitrogen and we have 2 hydrogens. Okay, so we've got a carboxyl end and an amino end. And then this is the R group, which is unique to each amino acid. So we have 2 amino acids here. So how do we form a peptide bond? It's actually a dehydration synthesis reaction. So these 2 things are going to combine together. So what does that mean? Look, OH and H, that's going to leave and form water. So we're going to break this bond here. We're going to break this bond here. And what's going to happen, these are going to combine. So how's that going to happen? So we're going to keep the nitrogen and hydrogen here. We're going to keep the double bond to oxygen here. These are going to leave as water. So H2O leaves. All right, so this is gone. Now, what does that mean? These are going to bond together. Now that line's not going to be that long because you come together, but that's going to be our peptide bond. Okay, that's going to be our peptide bond right there. So we're replacing this hydrogen bond to nitrogen with a carbon bond, and replacing the oxygen bond to carbon with a nitrogen bond, right? And they're going to share electrons and create a covalent bond. So then it's a synthesis reaction, specifically a dehydration synthesis reaction, and we know it's going to be anabolic, okay? And that's how you form this. This is the peptide bond. So peptide bond, and we just make this longer and longer and longer with all kinds of different orders and combinations of amino acids, because there's 20 of them, right? And that's our primary structure, that long strand of amino acids, right? And we make that with, the code for these proteins is in the DNA, to talk about nucleic acids a little bit. And, we form proteins from the DNA using RNA, which we'll talk about later, okay? So those are proteins, and just kind of give an overview of basically what we do with them in the body. Other thing, the denaturation of proteins, I would know that. So high temperatures, high or low pHs, and then they'll affect the structure of the protein, or even potentially salt concentrations, which would mean the osmolality or osmolarity of the body fluids could affect these, right? So we call it denaturation. And if these unfold, right, and you lose the structure of the proteins, they'll lose the function. So think of insulin as a protein hormone. It's bent and folded in a very specific way. it binds to an insulin receptor on the cell surface, which is also configured a very specific way for that lock and key fit, like we talked about with substrates and enzymes. And if that unfolds or gets damaged or is modified, the insulin receptor won't fit the insulin hormone, right? And insulin won't work, right? So just think about that as an example, and that's a big problem. Okay, those are proteins.